It's so nice to be, but I'm not too protected for the classes.
That's so
Do you say wear glasses?
I cannot see.
Okay, yeah, I was just like, you can't see that.
This is funny, but there's a gift beyond the kids' bed.
I feel that. I slept it happens like that. But you know what? My eyes are still working. And there's no growth.
That's the reason. So it's moving on. Good morning.
Match agents. I got in last night at midnight. Yeah, you're right. But yeah, I had a single sweet I really nice that one because that one's really fun. Yeah, I know.
First week.
I got a good day when I got back.
I did first two questions.
Great because I went back home for my mom's birthday and the Chinese New Year.
You better hurry it. Yeah, like,
She ended up moving most of our exams within years, like
So I
7
was
questions.
you seem sleepy.
Like she showed me so much.
Hey,
How's it?
you missed.
What? I finished. Jesus, you beat it to it. And then the so please. That's one of my favorite lines. Okay, we need to memorize. Hey, we're pretty close. We at least do.
After watching Spencer. That's around.
And then I just learned my class.
Why is there one?
But I still have to do work.
Yeah, I timed it perfectly.
Yeah, it's pretty early, but thankfully I already kind of answered the last one.
I'm about to
Wait,
Turn your machine on
so
one.
I think I just go straight to the library after this, grind it out, and read anything. Yes. Okay, so I might write a proposition. This is your proposition. I say after this, instead of doing that, you do actually and then you go to the library. Well, I might need to just grab something.
What? Oh, on like the softball time. What did you get?
Yeah, I just forgot to do that this morning, so I was like.
I converted.
That's
My
it.
bad.
I meant to, but.
Is that one we just read?
Yeah, it's just like
When's your part?
Today.
Yeah.
Like say you were in the.
So I was,
Which is your school.
So the Swanee record doesn't.
well, wherever I worked recently, it was common. But our first exam is not this Monday. How? Because on the listing, it says it's not, so I was like, oh no, we didn't. It was somewhere else.
Reese isn't coming today.
Oh,
she
she
hasn't Oh, because her toe.
Oh, she was on the right
Oh,
side.
what happened to her toe?
She broke her computer.
What did she do? She dropped a fridge on it. Yeah. Well, when you see her, tell her, I just followed her on Instagram, that's all I'm saying. Names.
I usually just send it, or just pause it.
Guess what? Oh, yikes.
That's
So
about
bad.
it. Last one.
I haven't noticed it on. Oh, that's what you were doing. I'm just not. I don't.
Alright, so this one, not gonna be a quiz question, it's gonna be more like a comments or feedback stuff, so do not put your name on these. That way you guys can be more honest about stuff. Today I'm gonna try switching up the class just a little bit. I'm gonna try this different way of presenting the data. And because I want the little cards to be anonymous, you can do that whether you like it or not. Normally, I'm always trying to tell you guys two different stories. Hey, there's really small properties, how they work, what are all these little cool little tools. But then I always kind of go to either these emergent properties, to figure out why this is important and work to work. Because I do both, even in my head when I'm talking to class, I feel like I go on these little tangents where I rant about what's so cool or how it works. It's interesting to me, and that's why I talk about it. But the problem is when I look at my notes back or look at the videos, I'm like, how would I write notes as a student? I wouldn't like this. So today what we're gonna try is, I'm gonna start super zoomed in. We'll look at the small stuff, right? So it's gonna be the first 20 minutes. I need you to be at your A game right now. Focus, good notes. Then you're gonna give me the last 20, 30 minutes, and there I'll go back and explain to you why it's important. At that point, I always want you guys to put your notes away, just listen and be like, oh, I think it's interesting, but I won't be thinking about the others. Yeah, so we'll try it out. If it works out great, cool. If it doesn't, I'll share the video from the morning class, and then you guys can back it up. All right, so let's do a little backstory, or backtrack to what we started with last week, right? So last week we were looking at the DNA. This guy, remember I was going so hard about it. This is our sugar, and it has different targets and different count. Then we have this other part, which is a phosphate, one which is that helps us create the table. The other one is we have these bases, which are called the nitrogenous bases. Which is one that basically can change the sequence that we have. So there, I want you guys to think about two different bases. One of them, here we're talking about nucleic acids, whether DNA or RNA. We're going to talk about how do I know where to start reading? Right? So for these, what we start reading is going to be our 5', and we stop reading at the 3'. So when we're looking at that, here in our DNA, right? There we can see there's our sugar. Remember, our first carbon is going to take the nitrogen base. Our next one is going to tell us whether it's RNA or DNA. Our third one is the one that connects to the following molecule. And then here, 4, 5, 5 is the one that we bring in with our phosphate. So when we're drawing it, it's like the front half, this is our phosphate group. Put a little chain to it. This is the hydroxyl group with the next one. To glue these, the next molecule would come in right here. And it would do this. It would put its phosphate there. And the chain would just build up like that, right? I'm not going to draw everything else just for the sake of space, but then we have a hydroxyl group left behind. And just like that, for me to keep growing, I'm going to take out this as water. I'll bring in the next phosphate group. And then we keep building these chains. Right? So these backbones are repetitive, and this is how we're making our cable. Right? This is going to be the covalent bonds, this thing's permanent. Then on this side, where we have our carbon 1 in the picture, is where we have the different nitrogenous bases, right? So those, remember, it's A, T, C, G if I'm talking about DNA, A, U, C, G if I'm talking about RNA. But we can switch those in and out, and those are the ones that are going to make the big difference of what we're actually reading, right? So A's and T's that you can see here, they'll give us these special bonds, but they also have these little extra molecules that are kind of be hanging out or sticking out from them. And those are places where other things come in and grab onto them. But this is where we bring in the variation, right? So everyone feeling pretty good about that one? Yeah? All right. So now we're moving up to the next level, which is what we're going to be talking about today, is going to be looking at amino acids, right? So amino acids, we're going to kind of look at them like this. You're kind of following the same thing. When we start talking about them, we're going to start looking at like the way that we're going to read them. The first thing we look at is going to be the N-terminus. And they call this because there's a H3N. So you see kind of like this. Right. Then we have, these will be the names for the amino acids. So then eventually you get to the last bond, which is, we have a carboxyl group, so they call that the C-terminus. Right. Sometimes they'll call them like amino terminus or carboxyl terminus, because that's what you have, the groups that are there, right? And as we're growing it, basically it's gonna be an amino group, there's a carbon, and then there's a carboxyl. When I'm bringing in the next amino acid, right, we're gonna do the exact same trick as before. We're gonna kick out some water, we'll bring in the nitrogen on this side, and what's gonna be left behind is gonna be, again, this is gonna be left behind, right? So this is gonna be here, HN, C, and then we have our CO2 hanging out at the back. Right? So that's the way that we're gonna be doing the cable. Let's go look at it actually like formula wise. Right. So here we're imagining, we're just following the cable now. This is the very front. There's our nitrogen group, right? Our N terminus. This is a single amino acid. There's your central carbon. And then here's your carboxyl group. So this is the front, this is basically the back. This would be a second amino acid. They're kind of giving us this R numbers, which represent what are the things that we can have in that pocket, and those things will change and they'll give us different properties. So we'll go a little bit over that a little bit later. So here what we're looking at is this reaction here, right? There's our O. Here we have extra hydrogens. We're going to steal those away, take out the water, right? So we got that dehydration reaction going on. Help out the water, and then there's going to be a new bond formed between this nitrogen connecting to that carbon. Right? And now you have a dimer. Amino acid 1, you've linked it up with this new connection. Now you have amino acid 2. And this back end looks identical to this one. So it means that you can literally just imagine this, you pop it back up here, and you can undergo this again. And that's how you can just keep making chains of add another amino acid, add another amino acid. Yeah? Everyone's feeling pretty good about that? All right, so that's the cable, right? Now we got to get into what is this R group? Like, what is this thing? For this, we're gonna go back to you guys' favorite stuff. Remember polarity, polar and nonpolar, right? This is where we're gonna start talking about that again. So for the amino acids, there's 20 of them, and I don't expect you to know how to draw these, right? I do want you guys to feel comfortable with the purple side, which is your N terminus, the central carbon, and your CO2. I do want you, because that's repetitive across most of them. Up here, the thing that I want you guys to kind of follow is, like for example, this first set, so these are non-polar groups. And for the most part, what do we see is a bunch of CHs, right? CHs all over the place. Remember, when we're drawing these lazy, if we put up just the lines, it means all these are carbons. And there's nothing there, it just means there's a bunch of hydrogen. Right? So there's a bunch of non-polar bonds, non-polar bonds, non-polar, non-polar, non-polar. So we imagine this, when we're building our chain, back to my little rough drawing, right? We're building a chain, you're gonna have these R groups showing up every so often. At the central carbon, there's gonna be another R group and so forth. Depending on what's popping up, which is the colorful thing here, it'll tell us how it'll behave, right? So if it's sticking out and it's non-polar, that thing is not gonna wanna work with water, and that's what all of these are gonna do, right? They're gonna be super uncomfortable hanging out with water, they'll try to go hang out with themselves or with something that has carbon with hydrogen, right? The next group, you're gonna see it's gonna be a little bit different. Now we're looking at the polar side chains, right? So for this ones, the things you're looking out for, look, there's your oxygens, there's your double bonded oxygens, there's your amine groups, oxygen, oxygen, right? OHs, remember, that's the one I want you guys to memorize. That's already coming in, that's gonna be the polar side. These things are gonna make a little dipole, and you have a little positive side and a little negative side. And that's gonna be the thing that's gonna attract this thing to water. Again, you don't need to memorize the colorful green part. Just feel comfortable that, so, hey, you start seeing these OHs. I'll skip this one because there's two S's on both, but we'll focus on these OHs. If you see OHs in your head, I want you to think, hey, that's a polar. And you already know this thing's gonna be hydrophilic and it's gonna stick to water. Yeah. Then the last group that we have is the ones that are gonna be charged. So for these, again, the purple thing is going to be exactly the same as before, N, C, C, so N, C, the carboxyl group. These are going to change. The main thing here is that, or the fastest way to find them is by looking at their charge, right? So you're going to see here, this NH, the N is really good at capturing hydrogens. Technically, an N would only need 3 partners for it to be like fulfilled, right? For it to be complete. because it has this carbon bond here, that's 1, and 3 H's, that means that has more partners underneath, that means it's sharing more of its electrons, and that's what gives it a positive charge in that region. So there's a positive charge there, there's a positive charge there. And then for that, what that means to us is that when this amino acid is present, you put it into a solution, what is it gonna do? It's gonna come in as a NH2, and it's gonna steal the hydrogen away from whatever's in that environment that it shows up to. When it does that, taking away hydrogen from a reaction means you're gonna make it more basic. So these will have basic properties. Here on this side, it's basically the opposite, right? Which is, you see those carboxyl groups, this oxygen is gonna go into the solution. It originally comes in with the OH, and what it's gonna do is it's gonna completely shred the electrons from the hydrogen and kick it off. At that point, you lose a proton, so that means now you have more negative than positive charge. And that'll give us this. And because you release an H on its own, you just make the thing more acidic. So acidic ones and basic ones. Everyone feeling pretty good about these? Yeah? All right. The bluing. So for this one, I would want you guys to make it a little bit nicer on your notes when we talk about how it looks, or what makes up an amino acid, right? So in my question, I put, what are the 4 corners to the alpha carbon? But I've seen in some quizzes, they love to ask you, what are the 5 components of an amino acid? Right? So it means that the 5, most people start talking here, right? That's your alpha carbon, this is your central one. This one gives us the 4 different groups that are attached to it. Our amino group, which tells us the front, our carboxyl group, which tells us the back, our R group, which will tell us is it polar, non-polar, acidic, or basic. And then on this side is just the hydrogen to kind of fill the octet. So these are the big 3 players, right? These will make you build a chain. That'll tell you what that chain can do. All right, any questions on amino acids? Everyone feeling pretty good? Feeling that it can make a cable, yeah. Feeling that it can make little properties by sticking different R groups up a little bit. Let me see. I think at this point we'll call this, let's do it pretty fast. Well, I did it in 12 minutes, cool. All right, that was the heavy part of the lecture, right? Now I'm gonna go back and I'm gonna talk about the light things, about how these things are actually coming together and working, why it's so important. But within that, if you guys can, trickle in some questions. If you're a little shy, don't wanna ask the question, just write it on your card. I'll read those and answer them before next class, yeah? All right, so let's go back. Let me show you guys next. All right, so my favorite thing, central dogma, right? So we normally go DNA. This this has all of our information, right? Huge long sequence. We jump down here to our RNA. Where only some snippets of that get read into this part. Then our RNA code is the one that gets made into our amino acids, right? This is what we're looking at here. Okay. When we're printing it, it almost feels like we're 3D printing it. Our body literally glues one amino acid at a time, doing this reaction over and over, right? That's an amino acid, moves to the next one, that's the next one. It just follows to make it whole protein. At that point, you're gonna have your first thing called like a peptide, which would mean that it's just, you're looking at just one single chain. There's one N group in the front, it's linked up to all the amino acids, and then boom, you get to the C group all the way to that. The proteins, the cool thing about them is that they're like little machines that can actually do jobs. So in my head, I almost think of them like, they can almost like, you can think about them like tool sets, right? Like your pliers, they have a certain shape, they have a certain grip, and depending on how you grab them, you can really like hold on to something and move it, and it does a specific job because of its shape. Like I wouldn't want to be trying to put in a screw with a set of pliers because those things don't work properly, right? Or same thing, I could put in a screw with a screwdriver, but I wouldn't want to be trying to do like plumbing with a screwdriver. Like it wouldn't work out. The proteins are going to do the same thing. So they'll build themselves from this first, they call like the first level, which is, it's just a cable. And then these colors, you can kind of imagine on how you're peppering in hydrophobic, hydrophilic. Like telling us, is this going to stick to water? It's going to stick to the plasma membrane on the inside? Or is this going to be acidic or basic, right? We're organizing that Normally, a good, well-made protein, what they do is when they fold on themselves, the regions that are hydrophobic, they tuck into the protein, right? And because they tuck into it, the hydrophobic parts hang out with each other, and the hydrophilic stuff, normally you keep it on the outside, and that's the thing that can go and make interactions happen, right? And that's when you will start seeing that these proteins can make bigger shapes, or they'll start doing like 3D structures. Some of them, they rely on like a group of proteins. So there's these other proteins called chaperones that actually help twist them into place. Or when like the hydrophobic part's coming out, they'll hold on to it with hydrophobic parts so the water doesn't mess it up. Once it's done folding, it lets it go and the thing will collapse onto it. And then you have a nice folded protein. Some of them actually need their partners, like it takes a group of them to actually do the work together. Right? And that's when you get these polypeptides. So I'm gonna get into a few of them throughout the class. In 233, or the one after this one, I get super into them and we'll actually chase the amino acids down to the sequences. And I have another class where we actually make some of them ourselves. All right, but let me go back. Here we go. So then you can imagine it's a cable, and then this cable has all these abilities to kind of coil on itself through spins, and you'll see stuff like this. So this is one way you can look at it. This one really shows you kind of how the atoms are distributed. And you can see it's not just one long strand like we always see with the DNA or RNA, but it's all kind of twisted into itself, right? So this is kind of the space filling model. The next one is when we look at this ribbon model, what they're actually keeping track of is they're kind of imagining this cable, but they're hiding the R groups from it, right? They're saying, how is this cable going to build itself? And then you're going to see cool things like this. So this means it's disorganized. And then you're going to have these little things that look like a little ribbon spinning in space. Those are called alpha helixes. And the cool thing about those is, normally what they do is, all their R groups are going to be polar. So they're going to want to interact with water. So what they'll do is they'll stick, like one of them, the first amino acid, they'll stick it polar out that way. As you go down the chain, the next one is going to stick it up to the other angle. And they'll basically spin like that, stabilizing themselves, where everything on the outside of this is going to have that polar bond, that thing where it wants to grab onto. Right? This is one of my favorite ways to look at them. It's a little harder. Normally I prefer to slice them a little bit more. But in here, the baby blue is actually showing you what the R group would look like. Right? So you can see in there, I know it's a little bit noisy, but you kind of can see things like this where this R group is hanging out. There's a little, probably a carbonyl group or an amino group kind of hanging up there. And because of that, it'll literally be able to match a specific part, right? So if the receptor is made and you think like, hey, it has a polar bond on top, non-polar in the middle, and a polar at the bottom, the thing that comes in has to come in as polar, non-polar, polar, otherwise it won't stick. And that specificity is the way that we know that one thing will bind to one receptor and it will not bind to anything else because that pattern isn't set up properly. So deep down, that's the thing inside, but the big thing is that that protein's giving us that super special shape. And then what we're going to do throughout the classes, I'm probably going to bring up examples and show you some of these as kind of as they work or show off like the cool things behind them. This one's a pretty cool one, right? So again, this one I'll put notes on. You don't need to memorize it at all, right? But what we're looking at here is this is basically your amino acid, right? So we have our own cable, it's folded into itself. And the cool thing, it has like a little pocket where it's made to grip a lactose. If you imagine a lactose coming in, what do you see, right? Your brains are seeing a bunch of OH groups, means this thing's gonna come in super polar. And once it's inside, this thing has the amino acids in basically this binding pocket that'll help recognize that shape and it'll be able to grab onto this thing. Right? So that'll be that special design, boom, it grabs to it. Then what it does, it has an active site. And through that active site, it'll temporarily break this bond, it'll help break the water and help reconnect them. Right? So one of the hydrogens, let's say this hydrogen, one of them will go into oxygen, giving us this. The leftover HO, someone's going to link up into this. Right? So it'll go in there, it'll help bring these two things together, and it'll bring in the water pool, and then now it can cut them into two pieces. If you put a different size sugar, right, like sugars, you can have them with these OHs where they're both up, or the H's can be down. 5-ring sugars, 7-ring sugars, remember all those fancy things? those won't fit in that pocket. So even if they're there, they wouldn't be able to jump right in and get cleaved because they're a different size, or their polar roots are in the wrong spot. So that's how this thing's perfectly being driven and cutting the thing that makes the grip. Jesus, that looks nasty, guys. That was weird. All right, so throughout the class, I'm gonna talk to you guys about a lot of these different ways where we actually see these proteins and where that shape is super, super important, right? So in this case, we're talking about, like, for example, there's hormonal proteins. And these are the stuff that help coordinate our entire body, right? In this case, we're basically releasing just insulin, that small little chain that goes, and it's gonna go and it's gonna bind to its own specific receptor. So a receptor would be that thing that's gonna go into it and grab it. And normally what happens is when it grabs the signal that it wants, it'll do an internal thing of, now it sends a message out to the rest of the cell, and it'll tell it turn genes on, turn genes off, mess with the protein, turn on a protein, whatever it needs to, it's gonna get passed down. Right? But the cool thing I think about this is, like, our cells are set up so nicely where, like, even if I'm the cell sending the signal to the whole class, only the ones with my receptor would be the ones that actually get the signal, right? So if I'm like a good example is right now, it's like most of you in the class are not receptive to Chloe, right? If I say Chloe, she instantly looks at me like, what's going on? Because she has the receptor for that, right? If you have the receptor for your name, if I say your name, I know you know exactly I'm talking to you. The cells can actually do this thing where they'll send messages, and depending on how they hear them, it'll be how they answer, right? So, like, if I was like yelling like line up, right, I might scare her instead of just if I was just calling it out. And the same thing, these things will be sensitive to your specific signal. They'll even be perceptive to how much of it you're sending to them, whether it's going to be a quick response, a slow response, or sometimes they'll even ignore the responses. But all of that is coming from these things having those amino acids in a really nice shape, and then the receptor on the cell that they're going to, again, having that really nice shape that can complement each other. This is when we're going to kind of go into this class a little bit. I promise I'll try not to go too deep into it when we present it, but it is super fun the way that it works. So this is we're talking about, like, muscles, right? You know, our bodies have these cables called actin, and you have this huge cable. It'll be a chain that's like, they're linked up, and you glue them to these, like, walls, basically. So this will be like the full wall. This will be like the full wall. These things right here, myosin, they're little motor proteins, and they're so cool because their body coils into this long cable. And then these things, they kind of look like feet on a centipede. And what they do is they burn ATP, and they'll literally change shape from doing this, and then they'll bend back like that. And because they do that so often, and they can all grab onto the actin, pull themselves forward, let go, and then keep grabbing themselves, this is literally how your muscles work, right? This thing, we send in calcium, it starts walking, and it'll move itself out. It'll bring the two walls together, and that's what brings in the muscles together. But that relies, again, actin has a special shape. These feet has to have a special shape to grab onto them. Also, they need, I'm gonna show you how they're gonna change this, how they change that curve is based on their binding partners. So in one cases, it'll be relaxed, and in the other one, it'll make them contract. So super precise. This one is fun. So our cells also make proteins that they kind of push around them to make their environment. And they're like little cables that kind of connect them, right? I'm really doing a really cool one right now where I'm talking I have a development class afternoon, and we're talking about how when you're being formed, your face actually like smashes into itself. Like it starts out flat, it curls up and goes into itself to give you like your mouth, your nose, like to seal up the plates, right? But in one of the cases, there's a plant hormone that kind of looks like the cable itself. So instead of cells being able to grab onto the cable and walk forward, they grab the chemical and then they don't slide forward. And when the face doesn't close properly, that's how giving that chemical to animals, you can get animals that become like cyclops, right? So their face is trying to close off, but because it can't move all the way forward, they end up just having one eye, like these parts basically blend into one single thing. And that's where this thing is super cool. You guys okay to watching something gruesome or no? Yeah? So that's the cyclops, right? Like the face is trying to close onto itself. And then this would be the left part of one eye, this is the right part, the other one coming in together, right? And it's just because that protein grabbing onto that cable for that cell, it uses to kind of pull itself forward. Because the chemical has a similar property to that little place where it's gonna grip, it grabs the cable, it doesn't grab the cable, it grabs that chemical and it holds onto it, and now it can't pull itself forward. And because of that, you can get some crazy things like this. Yeah, so it's super important, they're super critical. But again, this is just me telling you a bunch of stories, just to emphasize the thing of these proteins are gonna make a cable. Their sequence is gonna matter, right? Because it's gonna tell us what R groups we have present. Those R groups are gonna be set up in 3D space to actually be able to kind of fulfill a job. And they're super finicky and they're super picky. And this is just some examples of where it's like, it's been made to be super precise. Yeah, I think that's it for today, right? Questions?
Could you go back to the slide that has the different proteins?
Yeah.
The
Or the
bottom,
amino acids, right?
no, the different kinds of proteins. The bottom two don't have function, it's just kind of left blank.
Which two? Wait, this one?
The contractile and motor proteins and the structural proteins.
Yeah.
How would you, like, in a simpler term, I guess, say what the functions of those two are? It's kind of missing.
Contractile and motor proteins, right? Okay, so let me show you, let me get a nice picture. So at the smallest zoomed in level, the motor protein is a thing, right? That's what we care about. So it'll be each one of these little blue bumps is going to be your motor protein. That's going to do the thing where it's going to pull forward, grab onto the cable, and then it pulls itself. Like put itself kind of I can write something like this, like it's doing that, right? In that case, the shape of the motor protein, its ability to grab onto the action is super precise. That's the really pretty side about it. The cool thing is, now that we start zooming out and we see, hey, there's a bunch of these interactions happening locally, a bunch of proteins grabbing onto and coming forward, the big net effect is we kind of can zoom out and think about, this is basically, they're like, they're called little discs. And what's gonna happen is, if your muscle is relaxed, you can see these things are gonna be further away from each other, right? The moment we turn the system on, there's our energy, this is our signal. This thing's gonna start walking itself forward, and you're gonna see that the cables themselves are gonna come in. So this is really cool because these cables, they're almost like a chain, so you can't stretch them out, right? So when you're putting on them, whatever they're attached to, that thing's gonna be the thing that gets pulled in. Afterwards, you basically can bring in back the calcium. It's gonna stick to the little feet again, and you're gonna let go, and that's how you relax your muscle.
So would motor proteins only be in muscles, or are they anywhere else?
Oh, there's so many motor proteins, yeah, they're all over the place.
Okay.
Like this is just one example.
Okay.
Like there's some where it's like, when the cell's trying to walk, like when I was looking at this answer, the other one opened up by accident. I was like, no. But there's different proteins, and what they'll do is, you have the cell, imagine it's a big ball, and what it'll do, it has proteins that grip onto a certain cable or a certain strand. It'll kind of clamp into one spot. Then it puts cables that come out of that anchor, and it'll anchor somewhere else. And then what it does is, it'll start putting the cables together, and it'll let go of this one, and that's how it'll bring this thing forward. And it just keeps doing that. But it's a different set of proteins, they have different little clamps. But they're gonna do the exact same cool thing, like they grab onto their cable and they're gonna bend when we tell them to.
Thank you.
Any other questions? No, feeling pretty good? All right, let's call it then. All right, see you guys on Wednesday.